Aerobic respiration has been divided into number of enzymatic driven different processes like glycolysis, link reaction, Krebs cycle and electron transport system. This is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be talking about one of them, and that is glycolysis. Now you see, glycolysis is also known as EMP pathway, that is named after their discoverer, that is Embeden-Meyerhoff-Parnas pathway. It occurs in cytoplasm and it doesn't require oxygen. Okay, so here the pathway begins with a simple sugar. So even if there is a complex sugar, first of all it has to be broken into simple sugar in the form of C6H12O6, and then the next step begins. So ultimately the glucose is actually broken down into 2 molecules of pyruvic acid, that is a 3-carbon compound, and as a result 2 ATP and 2 NADH2 is formed. So this is a sequential steps. One by one we will talk about the different steps. Let's talk about the very first step. In the first step, what happens? In the presence of enzyme hexokinase, glucose gets converted into glucose 6-phosphate, right? Mind it, wherever there is a substrate level phosphorylation, then the enzyme used is kinase. Okay, so that's why here it is a hexokinase. In the step 2, what happens? That is a isomerase. The step 2 is a isomerization reactions, in which what happens, that glucose 6-phosphate gets converted into fructose 6-phosphate, right? So in the presence of enzyme isomerase, it gets converted into fructose 6-phosphate. The third step is very important, because here also 1 molecule of ATP is being utilized, and that utilization is also going to be catalyzed by one enzyme, what we call phosphofructokinase. So in the presence of phosphofructokinase, what happens? Fructose 6-phosphate is actually gets converted into fructose 1,6-diphosphate. Mind it, this is very important enzyme, because later we will discuss that how this enzyme plays important role in the regulation of the glycolysis. Now this fructose 1,6-diphosphate in the step 4 is actually broken down into 2 compounds of 3 carbon each. One is dihydroxyacetone phosphate and second is glyceraldehyde 3-phosphates. Even both of them are actually interconvertible in the presence of enzyme what we call isomerase, right? In the step 5, what happens? That glyceraldehyde 3-phosphate is actually gets converted into the 1,3-diphosphoglyceric acid. So mind it, here aldehyde is getting converted into acid, definitely hydrogen will be released, and that hydrogen is actually accepted by NAD, okay, and as a result NADH plus H plus is actually formed. So this is very, very important from that aspects, right? Now 1,3-diphosphoglyceric acid is actually converted into 3-phosphoglyceric acid, and again since it is a substrate level reaction, so that's why it is catalyzed by enzyme kinase. Okay, so the name of the enzyme what we call phosphoglycerokinase. Okay, now 3-phosphoglyceric acid gets converted into 2-phosphoglyceric acid. Okay, and that reaction is actually being catalyzed by phosphoglyceromutase, right? In step number 8, what we will see, that the 2-phosphoglyceric acid is actually getting converted into PEP or phosphoenol pyruvate. But mind it, this reaction is known for the release of a water molecule. Okay, and the enzyme that is utilized what we call enolase. Now this phosphoenol pyruvate in the final step, they gets converted into pyruvate, and as a result 1 molecule of ATP is again released. So here also the enzyme is actually kinase, what we call pyruvate kinase. So in that way we see our final product, the pyruvate is formed, right? Let's see the entire glycolysis in one diagram. Again you can see there that glucose is getting converted into glucose 6-phosphate, then it is a fructose 6 phosphate. It is broken down into 3-glyceraldehyde phosphate and DHAP. Now glyceraldehyde 3 phosphate gets converted into 1,3-diphosphoglyceric acid, then it is a 3-phosphoglyceric acid, then finally 2-phosphoglyceric acid, phosphoenol pyruvate and pyruvic acid. The important thing what you have to keep in mind, the place where ATP being consumed and the place where ATP being released. Mind it, this is one place where actually the ATP consumed, this is the another place where the ATP being consumed. But there are another two places where ATP is actually being released. Mind it, here it is released and here also actually it is released, right? So let's talk about energetics. So as far as the product of glycolysis is concerned, it is the 2 molecules of pyruvic acid, 2 ATP, 2 H2O molecule and 2 NADH2. Okay. And energetics we discuss in terms of what will happen in the absence of oxygen and what will happen in the presence of oxygen. Okay. For instance, if you talk about in the absence of oxygen, what will happen? The total number of ATP produced will be 4. Consumed is a 2. So net gain will be the 2 ATP. But in the presence of oxygen, what will happen? That the 2 NADH, what we see, that enters into electron transport system and each one is capable of forming 3 ATP. So altogether there is an additional 6 ATP can be formed, right? So what we can say that net gain is a 8 ATP in the presence of oxygen. So that is the two things what you have to keep in your mind, right? This is all about glycolysis, but before we end, let's see some of the problems which is actually figured in NEET examinations. So the question is, how many ATP will be formed through glycolysis by 1 molecule of DHAP? So mind it, here we are talking about 1 DHAP molecule that is going to be formed by the glycolytic pathway, and in that you have to consider the starting point as a DHAP, then only you can get the correct answer. Okay. So we know very well dihydroxyacetone phosphate, when it will enter, what will happen? What will the sequence of events? Just try to understand. The sequence of events will be that DHAP will get converted into 1,3-dipga. Here what we see, that 1 molecule of NADH2 is going to be released, right? 1,3-dipga gets converted into 3-pga. Here again 1 ATP is going to be released. 3-pga is going to convert it into 2-pga. 2-pga will get converted into PEP, and PEP will be converted into pyruvate. Okay. So here also there is a 1 ATP is actually being formed. So what is the total number of ATP? So this is 1, this is 2, and this is actually capable of forming the 3 ATP, right? Where? In electron transport system. Okay. So what is the correct answer? The correct answer is a 5, right? I hope you got the answer. So this is how you need to solve it. You can solve only when you go through the entire pathway, considering that one particular molecule, and with that molecule how many possible ATPs can be formed. That is a very popular question. Most of the time it is figured, but very easy if you just go through the cycle in a correct sequence. Okay. Let's discuss one more thing, that how actually glycolysis is being regulated. Okay. You see, glycolysis is regulated by enzyme called phosphofructokinase, and regulation is exercised through the allosteric sites, where what happens if there is a high level of ATP. So naturally the respiration would like to stop, isn't it? Because already there is a large number of ATP. So ATP binds to the allosteric site of the phosphofructokinase, and thereby the glycolysis is inhibited. Okay. So this is how the glycolysis is actually being regulated. Okay. Similarly, if there is going to have a ADP or adenosine monophosphates, so they will promote the glycolytic activity. Okay. So with the relative concentration of ATP and ADP within the cell, the enzyme, the phosphofructokinase, either it can be activated or deactivated. Accordingly, it will take care of the glycolytic cycle, right? Let's discuss one more thing, that is link reaction. You see, the pyruvate being formed, ultimately pyruvate will enter into the mitochondria. Okay. So once the pyruvate enter into mitochondria, then before it enter into Krebs cycle, it undergoes oxidative decarboxylation. So the link reaction is named because it is going to link the glycolysis with the Krebs cycle. Okay. So one thing, it occurs in mitochondrial matrix. 5 cofactors are required. What are they? They are lipoic acid, coenzyme A, magnesium ion, and thymine pyrophosphate. So these are 5 cofactors are actually required. Okay. So as a result of oxidative decarboxylation, what happens? That a 3-carbon compound pyruvate is actually getting converted into 2-carbon compound, that is acetyl coenzyme A. And in the process, what we see, that one molecule of NADH2 is also going to be formed. Okay. So one molecule of pyruvate is going to form one molecule of NADH2 and one molecule of acetyl coenzyme A. So this acetyl coenzyme A finally enter into the Krebs cycle. We will discuss the fate of acetyl coenzyme A in the subsequent class, right? And that is the Krebs cycle in the next session. See you there. Thank you.